Good afternoon and welcome to the second seminar in our National Myeloma series. I'd like to begin today by acknowledging the traditional custodians of country throughout Australia. I am lucky enough to be presenting today from the Dandenong Ranges in the land of the Wurundjeri people. We acknowledge their continuing connections to land, sea and community, and we pay our respects to elders past, present and emerging. We extend that respect to all Aboriginal and Torres Strait Islander people watching with us today. I'm so glad to be here with you all. My name is Laura and I am one of the myeloma specialist nurses with Myeloma Australia based in Victoria. I'm joined today by my co-host and fellow Victorian nurse, Kat, who will be chairing the second half of today's session. Peter from Queensland is assisting behind the scenes. March is International Myeloma Month, and in Australia during the month we aim to raise awareness of myeloma and provide access to experts in the field through these national online seminars and our local My Greatest Walk community events. These are just around the corner on the 30th of March. This year's seminars take place over 2 days. Our first was last Monday and today. Our theme, My Myeloma Matters, treating you, not only your myeloma, aims to take a deeper look at targeted treatments and individualised supportive care. We recognise that the many individual types and experiences of myeloma very much require a shift towards personalised medicine and support. Following on from Monday's seminar, we have some interesting presentations planned for today. On Monday, we learnt about how your myeloma fits within the current treatment landscape, your voice in shared decision-making, individualising exercise and all things stem cell transplant. Today, we will learn about the research and technology that drives today's new treatments, some highlights from international conferences that impact treatment and supportive care in Australia, as well as support of your emotional health, your relationships, and strengthening your immune system. Today's seminar runs for about 2.5 hours with a short 5-minute break around about 1:15 Victorian time. A reminder that today's session will be recorded and available for playback on our website. You can actually see the recording of Monday at the bottom of this page. The team will be monitoring your questions, which you can submit today via the live chat box, which is available directly below this screen online. We will do our best to respond to your questions today. You can also email any unanswered questions directly to nurses@myeloma.org.au. We would very much love to know what you think about today's session. Your feedback is important so that we can tailor future events to meet your needs. You'll find an evaluation link at the bottom of this webpage. It should only take a couple of minutes to complete. So with all that housekeeping out of the way, it is my absolute pleasure to welcome our first presenter. Associate Professor Edwin Hawkins is an immunologist and cancer biologist. He conducted his PhD at WEHI, which is the Walter and Eliza Hill Institute, before relocating to Peter Mac as a National Health and Medical Research Council Peter Doherty Fellow. In 2012, Edwin was awarded the European Haematology Young Investigator Fellowship to train in intravital microscopy at Imperial College in London. Edwin was recruited back to WEHI in October 2015 to bring his international experience to the Institute and establish a research program studying hematopoiesis, inflammation and cancer, which he continues to drive today. Thank you and welcome, Edwin.
Thanks so much for that introduction, Laura, and apologies for all the long, really annoying words in there. You did a really good job of them. But I think what I want to touch on is I'm going to show you some research today, which has been a passion of mine for a really long time. But as Laura outlined, I did always have in mind doing this project that I'm going to share with you today, even back when I was a lot younger. And this is the reason why I went to London. And what you're looking at in front of you is actually a picture of bone with big clusters of myeloma forming plasma cells in there. I'm going to tell you about this story today and how it relates to human disease. And I just want to highlight that the work that I'm showing today is from Joel Rhymes and Joy Liu, who are PhD students in the lab, Raymond Yip, who's a postdoc, and Jeremy Yu, who's a consultant hematologist at Peter Mac, who's now performing his PhD with me. So what we do in my laboratory is we find different ways of imaging cells in live tissue. We quite often do this using mouse models or preclinical models, and we find different ways of looking at tissue in action. And if you just look at the top left-hand picture that's moving around, that's an image I'm going to talk about a lot today. That's bone marrow. And why I want you to look at that right now is everything that you're looking at in that picture is happening in real time. So there's a big blood vessel, which is that black bit in the middle. The green and the red cells are cells that we've modified to make blood. They're the hematopoietic stem cells. And everything there that you're looking at is happening in real time, as it would in your body as well. So with this great technology, we can look at things that have always been really, really hard to understand, from culturing cells in a dish, and try to get a really good idea of how they function inside the body. So microenvironments is a really, really fancy terminology, and it's a concept that's not really well understood. So let me talk you through it. The idea is that in the bone marrow, where a lot of your hematopoietic cells live, and cells that drive myeloma, these plasma cells, that's where they live. We don't know how they work. We know that we get a range of different fates. Some stem cells will make blood, some will make immune cells. And the idea has been that there's little pockets and little areas, microenvironments, that have a unique composition of factors which drive this process. This has been a really interesting concept in blood cancers that have this intimate relationship with bone marrow, because the idea is maybe they move to these areas and they hijack them and take them over for their own good. The reason we want to understand it is we want to understand how certain therapies are failing in these patients, maybe because they form a microenvironment that stops an immunotherapy or a CAR T cell, but also just in general how it works to create an environment that these plasma cells that drive myeloma survive for a really long time. So the concepts at the bottom there, or how do they behave in the bone marrow? How do they hijack these microenvironments, and how do they modify them in their favor? So plasma cells are the thing that drives disease in myeloma, but also lupus. And they're formed from these immune responses that occur usually in your spleen and your lymph node. And then they migrate through a process that we don't understand to the bone marrow, where the fascinating thing about them is during an immune response, they're maintained there for your entire lifetime. So these plasma cells have sort of already evolved to be really long-lived. They'll survive for the lifetime of the individual in the bone marrow. Now, we know that they'll circulate through the blood and get there. And then when they hit these spots, that's a plasma cell drawn in the middle of bone marrow, there's osteoblasts and osteoclasts, and they make bone. And we know that the disruption of how they function is a really important thing in myeloma. We know that they disrupt the function of those cells, and that's why a lot of patients get fractures. We also know that they interact with the blood vessels. But there's a range of immune cells as well that have been proposed to be the things that help to support these plasma cells that survive for a long time. So, of course, the concept is we can figure out the cell type to delete and get rid of. Maybe then we can target these plasma cells better. So the question is there a niche or a microenvironment? Is it intrinsic versus extrinsic? And what that means is it genetic mutations that really drives this and how they function in plasma cells? Or is it this information from the cells around them? And then do they grow better? Do they survive better? Or is it a little bit of both together that combines over a really long time to cause the expansion of these cells? So here is a picture of what myeloma looks like in the bone marrow of an animal. This is a mouse model where we can mimic what happens in the mutations that drive this disease. We can transplant them into mice. And the left-hand picture is one of these TAL scans of the bone marrow of a live animal when we've transplanted in myeloma. And the really interesting thing about myeloma is it forms these distinct foci, these focal areas, where essentially the plasma cells will infiltrate, grow, and force everything else around them. But the right-hand side is a videotape, and people like to make these great videotapes of cells moving around in the body, and we do that a lot. And usually they're quite striking when you can see cells moving around. But the plasma cells are very different. They sit there, they form this foci, and they really do nothing. They just wobble around a little bit. And why that's interesting is on the left-hand side here, we've got some red cells that are acute lymphoblastic leukemias and acute myeloid leukemias, either with chemotherapy, without chemotherapy, with different types of drugs that change their interactions. And what I want you to look at on the far left-hand side is how fast these non-plasma cell blood cancers interact with the blood vessels and move throughout the bone marrow. So the fact that plasma cells do that is really, really unique. What we did is we made a really, really smart animal model where what we did is when one of the plasma cells, instead of being a healthy immune protecting plasma cell, becomes a myeloma cell, we targeted them so they would inherit a fluorescent protein. And what that means is then we can look at one cell that's a mutant and all of its progeny and understand what they're doing. So this picture shows it beautifully. You can see these individual clusters, huge clusters of plasma cells. What we usually see in somebody that's been immunized is somewhere between 3 and 5 plasma cells, and these are thousands. And what you'll see is a single color occupies a specific area. So what that tells you is this idea that a plasma cell can move to the bone marrow and make a microenvironment has a lot of strength from looking at it this way. So we know that that's absolutely true. And we've been able to repeat it a lot. The other thing that we know with these mice is they replicate a lot of things that we see in human disease, and that is it's a problem that we have with disease in older people. Mean age for diagnosis of myeloma is in your 60s. And these mice start to come down with myeloma symptoms about 550 days. That's quite old for a mouse. These mice will live for 2.5 years in the facilities. So these are really, really old mice, and that's what it looks like. But what we now know is that when we take young mice with this model of myeloma and we look in their bone marrow, despite the fact these are 80 to 100-day-old mice, so none of them will develop myeloma for another year, year and a half, they already have these plasma cell clusters in their bone marrow, and we never see these in other models of disease. So what it actually tells us is that these plasma cells move to the bone marrow in a mutated form really early in life, and they're maintained in these little microenvironments or niches there for an extremely long period of time. So it raises the question of when these cells that cause disease actually develop and move to your bone marrow. We now know from studies out of Iceland that when they profiled the entire population, well, about half the population, but they didn't select for whether people had disease or not. They just looked at their serum, and about 5% of people over 50 had an antibody profile like they had MGUS. So what it means is that we think that this shows that a lot of the understanding about when and how myeloma develops is actually not quite right. We think it happens very, very early on. But what it also tells us is that something isn't right about how we're thinking about these cells. So when they move into the bone marrow, the idea has been that they proliferate and grow really, really quickly. But I'm showing you data that says in really young mice, this clone will go there and it won't be a problem for another year and a half. So the nice thing that we were able to do is we were actually able to videotape this over a week in mice with myeloma, and that's shown at the left-hand side. Because we've got really dedicated graduate students, we then could randomize all the information to all the pictures and get them to count them. And then we could do that over a week because we can anesthetize mice and re-image the bone marrow and look at disease develop. And what we can see is that actually the turnover of these plasma cells over a week. So this is what my students like to call scientist week. It's Monday, Wednesday, Friday, so they don't have to come in the weekend, despite the fact they often do. But what we could see is that the rate that they grow is actually very, very slow. So on the bottom left here, we have the cell counts of the same population of clones over time, and it can show their turnover is really, really not that fast. One thing I want to seed in your heads today, and I'm not going to talk about it today, is everything that we found from myeloma so far, we also find for another plasma cell disease. It's called lupus, which is an autoimmune disease. Every single result that we find is the same between the two. So on the right-hand side, it's got plasma cell clusters and autoimmunity, them retained over time, and the same results. They're in microenvironments, they're clustered as a single clone, and their turnover is really, really slow. So what we did is we figured out a way that we would then image these cells inside a live animal, and that's shown at the top left, when they developed myeloma or lupus. We would then make a 3D map, which is in the bottom left of where all those clones were. And then what we could actually do is we could excise the tissue. Now, in mice, we use these landmarks to look at the marrow and identify it really well. But that also means it's very easy for us to cut out and isolate the cells from the area that we looked at and perform genetic analysis. So now what we're doing is we're looking at the RNA that they express, which is the message, and we can look at what's mutated and different in these cells. And what we found was something really, really interesting that I'm going to talk to you about, which is firstly, we hoped to get an idea of certain factors they made or proteins they express that would keep them in these microenvironments. And we actually couldn't find that. It was really, really hard to find. We found a bunch of control genes that we expected to see, and I've highlighted them here. On the left-hand side, that's the data for myeloma. Um, we see things like XBP1, which is a gene that's upregulated when these plasma cells are trying to make lots of antibody. It's a stress-related gene. We see overexpression of this MYC gene, which is commonly mutated in myelomas. And we see plasma cell genes like cyclin D2. When we compare these to normal plasma cells, we find about 74 genes are messed up, differentially expressed, is the way of saying it. When we look in lupus plasma cells, it's about 64. But the fascinating thing here, and the main thing I want to raise out of this, is that of these 64 genes that are differentially expressed in lupus, a completely unrelated disease to myeloma, 62 of them are shared between the two diseases. So what it tells us is that this interaction with bone marrow is probably evolutionary conserved. It's a function of plasma cells in general, which is really good because it probably means if we can figure out what's going on, we can probably broadly apply it to plasma cells in general. So we're very, very excited about that. The other thing is we found a bunch of genes which are called Ly6 genes. They're not very well understood. They code for a protein that is related to immune cells. They've reported to have many, many different functions, but that was really a red herring. And I'll show why it's interesting in a second. Now, one of the things that we're doing at the same time, and this will become apparent why it links all together soon, is we want to understand why these microenvironments form and how they talk to the normal immune system. So this videotape here is clusters of myeloma cells in green and CD8 T cells, which are your main immune cell, in red. And the question we're trying to ask is, how do these myelomas shut down T cell responses? And also, if we're to perform these experiments in the context of CAR T cells interacting with the BCMA antigen, which is the big one, ide-cel and cilta-cel now, or with bispecific antibodies. And what they are is an antibody that targets an antigen on the myelomas, and then they target an activating receptor, CD3, on T cells and activates them. We know the initial responses of patients are really, really good, but then after that, they tend to fail. And this is all work done by Jeremy, who's the consultant hematologist that I raised before. So what we could do, if the video plays, is we can ask this question about, well, what do myeloma cells do to exclude T cells? If they get in there, what do they do to shut down these responses? And how can we understand this better? And the idea is, when we know the genes that are miscoded, or when we know the function of this, we can change how we engineer CAR T or inform the clinicians to do it. But also we can get an idea of any other therapies that we can use to target this. So the first finding is really, really simple. And that is, let me just go back to that, that when we have these T cells in the context of now the myelomas, what you'll see in the bottom graph there, the bottom picture, is the dotted lines are where the myelomas are, and the red are all the T cells. And what we see is when the myeloma gets really, really infiltrated, it pushes all the T cells out. So that's not that unexpected. And the idea based on that has been these areas are immunosuppressive. They shut down the immune system because they probably exclude T cells from these areas. So what we can do now is we've partnered with Bristol Myers Squibb to get an antibody from them that is the same as the bispecific antibody that patients receive in the clinic. We can image the mice on the far left-hand side before they start therapy. On day 3, we can image when they're in the middle of this therapy and look at how the T cells function. And then we can look after day 7 at the relapsing cells. So it really does mimic quite well what we're seeing in patients. And the data that we saw is fascinating. So hopefully this video is going to start in a second. We can now look at the infiltration of these T cells and ask what's going on. So the first thing that people have said is, well, T cells won't be able to get into these areas very efficiently. And when they're in those areas with myeloma cells, they will get turned off in how they function. They won't be able to grow because the myeloma is so immunosuppressive. So we can videotape this in process. So Jeremy's actually interacting with the data. He's circling a few areas. On the right-hand side, we can see massive T cell infiltration in an area where the myeloma is pretty much being destroyed. And on the left-hand side, we can see really nice sort of area where the T cells are trying to interact with the tumor. And what Jeremy's going to do in a second is zoom in on a T cell in in action in these areas. And the first thing that we can see is you can actually see that T cell is proliferating. It's rounding up, dividing into two, and expanding. So the idea that these areas are immunosuppressive and shut down T cell activation and proliferation is completely incorrect. And that's important because we can reject any hypothesis or any work that we want to go into looking at around that. I think Jeremy's got a nice little picture of that in action on the side. You can see that T cell rolls up beautifully. And actually, it's very easy to see dividing T cells and immune cells in living tissue because they always have to stop. They can't migrate. They always have to become a big ball before they divide and then they race off. Okay, so I've told you then there's no problem. The T cells get in, they activate really well, they proliferate in these areas, there's no problem with it. So what is actually going on? And the finding here was quite striking and quite interesting. So day 3, day 4 of the response, when the T cells have started killing, we started videotaping them a lot, and what we noticed is this. So Jeremy's going to zoom up on a T cell in red here. It's surrounded by myeloma. He plays the video, and actually what was quite striking is you see that T cell basically explode and break up into these little fragments that it leaves behind. We've never seen that in any control or anything else that we've ever done before. So this is a unique property of these T cells in these myeloma clusters. So what we actually see, and what Jeremy's doing now is highlighting, actually you can see these red little puncta accumulate throughout the myeloma areas. And what that tells you is that we think the mechanism here is that the myeloma is able to secrete a product that shuts down the T cell responses by inducing death. We don't know if that's activation-induced cell death, anergy, exhaustion, but this really changed our thinking about the theory about how it might work. So of course we wanted to understand that better, but it's a really, really tricky thing to understand. So I've summarized it this way. Scientific data is a big thing of Legos. Cell biologists will like to tell you the different types you have and what they mean. People that do bioinformatics can look at all the gene expression and they can stack up all this data and quantify it. And when we do imaging like I do, we try to take the small amount of Lego bricks, the number of colors that we have to make it work, and kind of show you a picture of how it works. The problem is that we can only look at about 2 to 7 colors at the same time, whereas we've got 27,000 genes to look at. So we've always kind of had this approach of building these houses and understanding it, but we do a terrible job of it. So the way that we're addressing it is this. It's called spatial transcriptomics. And what we can do now is we can take tissue, we can image it and understand where clones are, we can understand where the immune cells are, or anything we want to do, and then profile the gene expression in place. Rather than stripping cells out and looking at what genes they express, like I showed you before for our sequencing method, we can look at it in place. So here we have a bone from one of these mice that have myeloma. We can see the different patches of the different clones. And then we can look at all the genes that are expressed in these places. And actually, what we started to see is this, and this is a fascinating finding. So let's just look at this left-hand graph. The idea was that every myeloma cell would remodel the bone marrow in the same way. So there'd be a consistent bunch of cells that they needed to work with to make these microenvironments, probably driven by growth factors that they make and other things. But we never found that. So what we find on the left-hand side is when you look at this Ly6K gene that I was talking about before, it's actually clonally inherited by different plasma cells. So it tells you right off the bat that they're very, very different in the genes they express. But underneath them, we're looking at the profiling of genes that are expressed in the bone marrow tissue itself, not the plasma cells. Cadherin 5, which is expressed on blood vessels, PLVAP, which is also expressed on blood vessels, but other markers for fibroblasts and things. And what we find is that every single one of these areas that have plasma cells is extremely different in the genes that they express. So that was not what we expected, but we're trying to resolve that with other data that we're getting from humans now to understand the question better. The problem with that data is it doesn't have the resolution that we want to have. So I'm really happy to share with you some new data that we've got, and we've applied this all on human. I'm going to share this data with you now of this new technique, which is called Xenium. And we use this to profile the genes in place. So that was an H&E section of bone marrow. I'm clicking on genes to mark bone and blood vessels, which you can see now from this overlay of the morphology, and it works really, really well. And what I'm going to do is highlight now how much resolution we have to understand the genes that are differentially expressed throughout the marrow. So I'm building through, and hematologists love this because it gives a really good idea of all the distribution of the cell types we've always wanted to know. So blood cells, immune cells, stromal cells. Here's megakaryocytes that make blood. They look absolutely gorgeous. Neutrophils as well. So we have this technology working really, really well. We've just applied it on a bunch of samples that we've got from patients as well. And I think that's what I want to talk about now. So we can profile the genes expressed. We can take samples from patients at different stages of myeloma, smoldering myeloma, MGUS, newly diagnosed, relapsed refractory, and start to look at what the commonalities of the genes are expressed are. So here we have a trophine from our amazing patients that are involved in these studies that donate tissue. And what we can do is profile all the different genes that are differently expressed in the plasma cells, but also the stroma. And we can also do it for mouse, but let's not talk about mouse anymore. So this work was all driven by Jeremy Raymond Yip, who did all the spatial work, and then Andy Chen, who did the analysis. We set up a way of now taking huge cohorts of patients. So we've got about 300 trophines. We've only assayed 21 of them for now. Make sure we've got great trophines on these patients. We can then go and look at the genes that are differentially expressed and understand what's going on. And here is a set of data that we've got. As I said, we've got 21 of them now. But all this purple and yellow coloring that you can see here, these are the fluorescent probes to detect all of these genes. So we've got it working really, really well. And this is what the data looks like. And this absolutely blows my mind for someone that's worked on bone for an immense amount of time. This is a control sample from a patient who has follicular lymphoma with no bone marrow involvement. And what we're doing now is dumping every single gene transcript into this sample. 5,000 genes we're looking at, and then we can build up this great encyclopedia of where every cell is, what they're doing, what they're interacting with, and what it looks like. So I'm just going to show you this for a few samples. As I said, we've got larger cohorts of patients now, but you can see the teal are the plasma cells. So at the top are control samples, MGUS, smoldering myeloma, and that little teal population there, that is the plasma cells. And then when we look at newly diagnosed patients, you can see this massive expansion of plasma cells, as we'd expect. We can then look at genes that are differentially expressed between the two. And the outline of the red line there shows this massive expansion of these plasma cells from early stage of disease to late stage of disease. But then we can map them spatially onto these trophines, which is what's on the bottom with the diagnosis standing, CD138. We can look at where all the plasma cells are. And I like this one, MM3 here, because it shows a nice little purple blue area where we actually have the red blood cells that have survived because they're not too infiltrated, and these two distal ends with a lot of stuff going on. And the big finding that we've had is that everything that we see in mouse, we see in human. And I'm going to show it here talking to this trophine that we're looking at. We can look at the relationship of the plasma cells and where they're on the trophine. We can find all the different cell types. And then we can perform a gene expression analysis to look at what different clones might be doing, where they might be located, and then what's on around them. And what you can see is this red and blue clone that we can find at these different areas of the trophine. And the genes that they express are so different, and their microenvironments are different. And why we think that's important is we think some of those areas are the ones that are causing CAR T cells and people having bispecific antibody therapy. We think that they're completely turning off those T cell responses. And we're about to do a big trial looking at 50 patients before and after CAR T, 40 patients before and after bispecific antibody to understand this. So it's a really, really exciting time. I'm going to finish there. So we know that these plasma cells have this really interesting interaction with the bone marrow. We know it's completely unique. We know that every single plasma cell clone probably has a different microenvironment and not a conserved microenvironment. And a lot of people were disappointed when we talked about this because they said they kind of lost hope and understanding if we could target a certain cell type that was part of that. But upon reflection, I think it really does model what's going on disease a lot closer, and it's a problem that's a lot easier to understand. I think certain plasma cell clones definitely make different factors. They model the microenvironments around them. Some of those are permissive to immune cells, some aren't. And I think actually understanding that might be key for understanding immunotherapy and where we go to next. And as I said, we're about to do a bunch of new spatial work with cohorts of patients, common oncogenic drivers to understand how certain gene mutations drive these microenvironments. And I think it's going to be really, really interesting in the context of these therapies. So I just want to finish by thanking all the people involved. I do get on the microscope a little bit, but nowhere near as much as the people in my lab. But just highlight the work that you've seen today was done by Julie Yu and Joel for all the original imaging studies. Jeremy has done all the bispecific antibody steroid studies in mice and all the human work as well. And all the spatial work is done by Raymond Yip, who I collaborate with, Rory Bowden and Kelly Rogers. And finally, we have great collaborations with Peter Mack. Simon Harrison has co-supervised multiple students in my lab. And that's been really key for us to make sure our results are relevant for patients with myeloma. So thanks very much, Laura. I'm happy to take questions.
Thank you so much, Edwin. The visualizing all of those amazing clonal changes is just, often with myeloma, we often say, it's hard to understand what's going on because there's nothing to visualize necessarily unless you've got bone disease or something. So it's wonderful to see that.
Thanks. And I think, Laura, I'd like to also add that now we've got it to the point where we can transplant myeloma cells from patients into a special type of mouse, and then actually add an antibody in and image their plasma cells in bone marrow. And I think that's the next step, is understanding certain mutations and how they drive that behavior. So the links there to what's going on in human are very, very close.
It's so exciting. I'm imagining a future of, everyone being gene profiled and us looking at your amazing work and having that to really individualize the treatment choices that we actually choose for these patients. Quick question, and this is probably a little bit simple, but we do know that there are some clonal changes already in myeloma that do drive the decision to treat with one treatment versus another, or might help us risk stratify a patient. Can you elaborate a little bit on what we already know and how that's actually creating treatment decisions in the Australian community at the moment?
Well, I think if I can be bold and take the next extension of that. So, Laura, yeah, absolutely, we know the association of certain oncogenic drivers and what they do. And Laura's 100% right. That changes how they're treated. I think what we want to do now, and I alluded to this before, is perform our work with really good representative numbers from each of those mutations, and then try and understand if they have a common outcome in those microenvironments for every single one of them. So, for instance, you might understand that certain oncogenic drivers drive completely immune permissive niches or immunosuppressive niches. So those patients might need completely different therapy in the context of immunotherapy. Or the other one might be that those common oncogenic drivers give such a diverse range of microenvironments, it's not something that we have to worry about at all. So that's what we're thinking about. And we're trying to get funding now to do those sorts of studies. Luckily, because Jeremy is so dedicated to this work, we have this amazing resource of these biobanks of trophines that are donated by patients. They're prepared in a way that we can do spatial on them. So I think we're not far away from asking those questions. So that's what I would say in the context of that question.
Oh, it's so exciting looking ahead and moving past the standard of care and more towards hopefully a bit more tailored choices depending on what we actually know about each patient's disease. Amazing. Thank you so much for your time. I imagine there might be some people who will ponder what you've said and send us some questions, so we will be in touch. Otherwise, thank you again for donating your time and for your wonderful presentation.
Thank you so much for having me. I really appreciate it. And I really look forward to questions. Please feel free to reach out.
Wonderful. Now, I have the pleasure of introducing our next speaker today, a fellow nurse. Please say hello to Jackie Jagger. Jackie is a haematology nurse practitioner. She's currently supporting the Central Coast Local Health District in Wyong and in Gosford in New South Wales. She's a registered nurse with over 30 years experience in haematology. She's also a committee member of the HSANZ, which is the Nurses and Haematology Australian New Zealand Society, and part of the Myeloma Special Practice Network as well. She has led and co-led a number of myeloma clinical trials and also teaches postgraduate nursing programs. She's developed several nurse-led programs and models of care which aim to transition care from the inpatient to the outpatient setting, which we know is so important and has huge impacts on quality of life. Jackie is a committed advocate of patient self-management and wellbeing, and we are lucky to have her join us today to share her insights on local and international research which impacts care here in Australia. Welcome, Jackie.
Thank you very much, Laura, for that kind introduction. I feel like I've almost got to live up to it now. Oh my gosh. Oh my gosh.
That's really you.
I'm just going to give a disclaimer before I start because I've been quite sick and still not great. So I'm going to be sipping lots and blowing noses. So please just ignore me and hopefully you understand everything I say. I'm very grateful for the opportunity to speak at the myeloma webinar. Fantastic that so many people get opportunity to see such level of education. I'm going to bring it down a touch, don't you worry. That was just a fantastic presentation by Edwin there. I was very, very lucky to go to Rio last year for IMS, which is the International Myeloma Society annual meeting, which has all the movers and shakers of myeloma there and all the people whose research I've been reading for over the last 30 years. So very, very lucky to participate in that. And I'm just going to share with you some of the key things that I went to. So mine was maybe a bit more from a nursing focus and from the focus of working in a regional setting. So I don't really speak about sort of CAR-T immunotherapy. It's probably more around access and toxicity management. Oh, remember my clicker. So the common themes that the sessions I went to were around bone health. There was a lot around exercise. I'm actually not going to speak about exercise. I've taken my slides out because there was a fabulous talk from Brent Cunningham on Monday, which I think will be able to be accessed for people who went at that meeting. It was absolutely fabulous and covered anything that I was going to say, so I've taken that out. We'll look a little bit at individualizing therapy, not from the perspective of the prior talk, but more from the perspective of dosing and reducing toxicity and symptom burden. Steroid toxicity is always topical, and I think we all strive to do something about steroid toxicity. We'll talk about some models of care. We certainly in our arena, we've had a 30% increase in patients coming through our cancer day unit every year for the last 5 years. So we are not by any means unusual. Many of those are patients who live with myeloma and now live long with myeloma, which is fabulous. But it does mean that we've got lots more patients in the clinics and we've got to find better ways of delivering care. I've got a couple of slides on treatment of relapsed patients in resource-restricted regions, which, do you know, we are classified as a resource-restricted region. I think when you compare us all to the US, which may be resource-restricted, but probably is more equitable. And long-term therapy, looking at particularly maintenance lenalidomide. So my first couple of slides. So the International Myeloma Society meeting is over a number of days. And the first day of that, we also have the Nursing and Allied Health Symposium concurrently. I was lucky enough to be a part of the faculty this year. And our first session was a session called Moving Forward with Bone Health. And we had the fabulous Jens Hillengas, who is a doctor from Roswell Park Comprehensive Cancer Center in New York. And he was talking around contemporary understanding of myeloma bone disease and its management. And was talking quite a lot about the differing imaging techniques. So I just wanted to sort of, it's going a little bit back to basics, but talking about the decisions around which imaging techniques people are using. So conventional X-rays are okay for looking at myeloma lesions, lytic lesions in, say, for example, the skull. I've got an image here in the long bones. But they've got limited specificity and sensitivity. So that essentially means that their ability to test, measure, correctly identify myeloma and do that all the time and not pick up something else that isn't myeloma is limited. So although they certainly still have a place, X-rays most definitely do have a place in imaging in myeloma, there are better tests for different things. So they're good for long bones, good for skull. CT scans, so computerized tomography, they're the X-rays that are taken at 360 degrees, and those beams make up a 3D image. And the computer does fancy things and gives us the slices. You can see here on the third image, you can see all these slices. So cross-sectional slices of the body. And that is a much higher sensitivity and specificity than X-rays. and it will detect about 25% more bone lesions than a standard X-ray. CTs can also tell us if there's infiltration in the long bone, so it can detect if there's infiltration of the soft tissue in normal fatty bone marrow. So we all have fatty bone marrow, and we all get a bit more as we get older, and it can detect whether there's infiltration of disease there, but more so in long bones, and not when, certainly can't see when we're looking at things like the spine. So then we go on to things like the MRI and PET. So the MRI and PET can provide high sensitivity, especially in earlier stages of disease. Infiltration patterns of the bone marrow are often diffuse, so spread around in the bone marrow in myeloma. But Jens found in one of his collaborative studies, he's been the author of a number of studies, that the infiltration patterns differed a little bit more so than they expected. So they found a third of patients had this diffuse pattern of disease in the bone marrow. A third had focal lesions, and a third had both this diffuse and local infiltration in the marrow. And these are important things to understand, because the number of lesions can have prognostic significance, so can mean provide more information about how we expect somebody's myeloma will act, whether it's going to be a naughty one or not. And Jens is also undertaking study into the biology of these infiltration patterns, which was really interesting, looking at the genetic background of the lesions with CT-guided biopsies into some of the focal lesions. The limitations of the MRI really are that it can be difficult to differentiate between lesions, bone lesions, anything in the marrow that's still active, or whether there's just scarring. So that's where PET-CT can help, because PET-CT uses glucose taken up by active cells to help us understand the metabolic activity. And that correlates very nicely with myeloma disease activity. So just differing choices of myeloma bone disease imaging. And I think the broad stroke of that is that CT and X-ray are good for hard bones, for mineralized bones, and CT is better than X-ray, and PET and MRI is better to demonstrate bone marrow infiltration. MRI is also done not just for bone marrow infiltration, obviously, but we do very much so when we've got spinal disease, just to better understand how the bones are affected, and any soft tissue infiltration into the bone marrow. This slide here, just the images on the bottom demonstrate that. They've actually zoomed up, so yours, I don't think you can quite see, but it's just trying to show really, these images are from Dieberg and colleagues, and they nicely demonstrate the differences between that sensitivity and specificity. So the first image is a plain X-ray. The second one is of second and the third are PET-CT, and all of these fail to pick up a lesion at C7 at the cervical spine level 7, but they are picked up on all of those MRI imaging. So I think it's, again, just speaks to the fact that MRI is very good for spinal infiltration. So people will often say to us, so why aren't we all having MRI? Well, you know, a number of reasons for that. It's very expensive, it's not easily accessible, and it doesn't, most people do not require MRIs to accurately assess their myeloma bone disease. It's more related to when people have got this infiltration patterns in the bone marrow, and if we've got spinal disease that we really need to understand a little bit better. I'm then going to flick over to somebody many of you may know. So this is Dr. Tracy King. This is a very good friend of mine and a colleague who we've worked together for decades, both lovers of myeloma. She's a myeloma CNC at RPA, and she's a doctor because she's completed her PhD on the development of a steroid symptom questionnaire. Many of you in the audience may well have participated on this study. She presented in Rio the validation study, so it's the phase 4 of her big body of work for her PhD of the steroid symptom questionnaire. This is it at the bottom, and the concurrent symptom of life measurement. So Tracy, for any of you who know her, me, people who know me, we're fervent supporters of trying to better understand the symptom burden from steroids, and I'm constantly badgering and constantly bring down doses of steroids at appropriate times to try and reduce the impact on life. So the PhD was really the development and evaluation of this patient-reported outcome measure, so PROMs. There's many of these around now. There's never been a patient-reported outcome measure for steroid in myeloma. This was adapted actually from steroid in a different setting. So a really important, I think, assessment tool for a very, very common problem in myeloma. And it's designed to assess the impact of steroids associated with therapy for myeloma. It was tested in 5 hospitals through New South Wales. With a sample size of 140 patients, and we asked patients to describe the incidence and severity of the steroid-related side effects over the previous 7 days, and that was done in 2 time points to try and ensure that we had valid answers and reproducible answers. So there's 20 items in the questionnaire. 19 are symptoms based, and there's one asking about people's blood sugar, of which most people didn't know whether they had a high blood sugar or not. It was quite interesting. And the higher total scores, so the higher SSQ score equals higher symptom burden. What I really like about the questionnaire she's developed is that we've got this option to list other. So if there's something that's a problem for somebody, they can list other, and also most bothersome. So many patients would have marked lots of different symptoms related to steroids, but we sort of pushed patients to say, okay, so you've got all these symptoms you're reporting, which ones actually are the most bother to you? So that's actually quite helpful for when you're seeing say, for example, the best case scenario is you do one of these before going in to see a hematologist, and they can pull the wood from the trees of, okay, we're not going to be able to address everything with this appointment, but what's the big hitters? What are the things we really need to try and sort out? Here's the one-pager of results, patient information on the left, and then on the right, the clinical characteristics. So I'm just going to do one more. Oops, sorry. Just looking for some highlighting. The patient group is what we see reflective of myeloma. So primarily male. The mean age was nearly 70 years with a range up to 89. So we had 89-year-olds on the study. That was one of mine. Most people had English as the first language, 91%. And interestingly, if we look at the range, the years since diagnosis, we had people from newly diagnosed to have had myeloma for 22 years. I think this is great and really encouraging. We see so many more long livers now, which is fabulous. And this, I no doubt, will get pushed out now. The mean years since diagnosis was 4.5, and most people had had around 4 lines of therapy prior to completing this study. So I think the big information in this session is this steroid doses. So the steroid dose at the top is the milligrams per week, and as you can see, it's not for the faint-hearted. We've got between 4 and 80 milligrams a week, and in a month that's between 12 milligrams and 180 milligrams. So huge doses of a drug that's actually quite toxic. So hence why we see so many problems with this. Interestingly, on the next line, we can see somebody's had 62 lines of that current regimen. So again, speaks to the fact that people are on therapy for a long time. And this is a long time on therapy that is not symptom-free. The main regimes, the top 3 were the len-and-bort, lenalidomide-based, and bortezomib and dara. And our ECOG, which is the Eastern Cooperative Oncology Group performance status. So that's a performance status that determines a patient's ability to tolerate therapies in serious illness. So 0 is essentially a fully active person who can carry on all the previous things before they got myeloma or started therapy. Grade 1 is restricted in strenuous activity, but otherwise can potter around the house, do light housework. 2 is up and about, out of bed for at least 50% of the day, but not working. So we stopped it there because we thought these were people who are typical of an outpatient unit, you know, a mobile outpatient unit. And we didn't want to get too caught up with other symptoms related to steroid. This is the only other slide for steroids, but this nicely depicts the frequency and severity of symptoms that we've seen in the study. So as you can see, a high frequency of symptoms, disturbed sleep being the biggest hitter and disturbed sleep being the most severe. So lots and lots of symptoms. And this shows evidence of acceptability of the questionnaire because all items were endorsed by patients apart from hiccups. And we kept hiccups in because even though it's not really frequent, if it happens and it's a problem, it can be a big problem. We've all had admissions of patients with intractable, so hiccups that can't be stopped. So we kept that in. But it just demonstrates that range of physical, emotional, behavioural issues. And it's a really pertinent reminder to us all that these symptoms just relate to the steroid. You guys are on regimes that contain, 2 to 3 other drugs, which have their own other symptom burden, or even if it's just Len has its own symptoms, my gosh. So it just speaks to the fact of the high symptom burden that patients with myeloma live with. I'd next like to flick to this god of myeloma. I have a professional crush on Dr. Vincent Rajkumar, partly because of his brain. It is amazing. I've been reading his papers ever since I started in hematology. But also he has just such a very clear idea of needing to apply what we know about myeloma, what we've learned from clinical research, to the individual in front of you. And that's really what he was talking to us about. So he was talking around individualization of therapy, and that is the role of the clinician. So what he means by that is when, say, in the Australian context, when we get a new drug on the PBS, the drug has been approved on the PBS on the basis of a clinical trial, what we call a registration trial. Often the trials are using quite high doses of steroid. They might have patients excluded. They might have patients excluded with systems problems such as, kidney problems, with liver problems, maybe patients who have had previous cancers, for example, or other comorbid conditions, other problems with their health that exclude them from the trial. But that's actually not the real world that we treat. The real world that we treat is people with lots of issues. So he talks very much around that. I'm not saying that we use drugs off-label, but it's absolutely fundamental that you appropriately dose the drug according to the patient in front of you. So that's sort of his mantra. He's very good at that. We were very lucky to get Vincent Rajkumar to the Allied Health and Nursing Symposium. Steroids, less is more, again, had the same approach as we do, and pointed out some of the studies that are coming through looking at steroid-free, and that should just say steroid-free regimens rather than therapies. So some cycles and regimens of treatment that don't include steroid or have very minor, and often these studies will be more maintenance because steroids are very important, particularly at new diagnosis and progression. And certainly we're not trying to get rid of steroids. It's maybe just appropriately dose as we go further down the track. Talked again about considering cycle schedules, dosing and length. So by that sort of, we've moved, bortezomib is a good example. It was intravenous, came down to subcutaneous to reduce the peripheral neuropathy, went from twice weekly to weekly for the most part, again, to reduce the neuropathy and other toxicities. Dr. Rajkumar gave examples of some of his patients who are quite old and frail that might have a dose of bortezomib every 2 to 3 weeks. So really about, okay, this patient in front of me, what can I do, and how can I keep someone on therapy that is effective for their myeloma, but not too toxic? So very, very interesting guide to listen to. And lastly, on this slide, just to, I've only done one slide on this god of cancer, but the importance of assessing frailty, particularly in older patients. And MSAG, our Myeloma Scientific Advisory Group in Australia, recommends the International Myeloma Working Group Frailty Index as the instrument of choice for assessing frailty in patients. However, it acknowledges that that is a bit of an unwieldy beast to try, and it's a tool that takes a lot of time, quite a few resources. And in our sort of resource poor centres, it was not feasible. So there's now a simplified frailty score that's an alternative, more convenient assessment tool, and it can be used in day-to-day practice in busy clinics. So that's a really good move forward to start better assessing people's frailty. Also in the real world solutions session, I was very lucky to be asked to be part of a panel, and the multidisciplinary panel was asked to look at models of care to meet this increased capacity requirement in the clinic. Other people on the panel were the fabulous Andrea Preston, who's a whiz-bang consultant pharmacist from the UK, Mary Steinbach, who's a myeloma CNC from Utah, and Dr. Rahul Banerjee from the Fred Hutch Cancer Center in Seattle. We were asked to look at strategies to manage the increasing number of patients and to look at how we balance disease and treatment burden. So the discussion centered on things like the route of administration. So we've touched on that a little bit already. So subcutaneous administration over intravenous, maybe oral over subcutaneous. Obviously, that depends on what drug you are having and what is the method of administration. But where possible, we try and move towards things that are not so cancer center intensive or necessary. And looking at treatment closer to home, so there was lots of conversations around hybrid models. So obviously in Australia, we've got perfect examples of the challenges for rural and remote patients, regional patients who are quite some distance from the centres, and accessing specialist cancer services. So we were talking about hybrid models where people are sort of starting to look and work towards working with the GP, the primary healthcare community nurses, community outreach, hospital in the home programs, and also looking at self-administration models. And I've got one slide with mine there to talk about next. The importance of prioritising life goals. So gone are the days. I mean, I'm old enough in myeloma to, you had your induction treatment, you had your transplant, and then you didn't have anything, before any maintenance, before any thalidomide or anything, which had its benefits. People had a treatment-free period, and they stayed off treatment until the myeloma relapsed. Not so good for management of the myeloma, and we've moved away from people being off therapy. So we now need to look very much so, how do we schedule, how do we choose the correct drug, and always have an eye to the toxicity burden of the therapy we're about to use to make living comfortable, enjoyable, and able to achieve the goals. I do, as a clinician, feel very strongly around the importance of the hematology, the myeloma clinicians taking responsibility as part of the promoting normal well-being, outside of your myeloma, because people are so often, patients are so consumed with just trying to manage the myeloma, the treatment, the burden of disease and appointments. So it's important for us to push forward those conversations around good cardiovascular health, respiratory issues, ensuring that we're trying to address obesity. Our drugs cause quite a problem there with steroid, exercise, bone health, vaccinations, etc. I keep wanting to press the wrong button, not the clicker. Here's just a screenshot of the poster I presented in Brazil, And this was of SLAM-B, which is self-administration of subcutaneous bortezomib in the home setting. This was in regional Australia, so in the Central Coast. I've just got one or two slides. This is the study design. So SLAM-B, all eligible patients were anybody with myeloma who was on bortezomib and who was willing to undertake some training, had the dexterity required to give an injection. That could be the carer. Most importantly, clinically stable. That was where we sort of fell down quite a bit. Often patients were unwell for quite some time. And people have to have history of obeying the rules, essentially, and not just going off on a tangent. The intervention was patients and or their carer, whoever was going to deliver the injections, or both people, and they would have one full cycle of education and training. Throughout that cycle, they would be, hopefully by the end, deemed competent with their injections. So supervised injections, they need to be competent for 3. Once competent, they could then move to the self-administration model, which means patients will just come in for day 1 of their therapy. They would give their own bortezomib in the day unit. We could monitor the technique just to ensure that was ticking along nicely, no issues. They'd have any intravenous drugs, the bisphosphonate or IVIG, etc. Pick up any orals such as Len, steroids, anything they needed for the cycle. Pick up the myeloma treatment scheduler, the tick box for the medications, and then they would deliver the rest of the regime at home. We did built into the design that we had pre-chemo telehealth assessments, because I was absolutely adamant that people would not miss out on education and support that went alongside those appointments for treatment. So it gave people opportunity to say, I've seen this, I've asked that, what's this, what's that, this is happening, is that okay? So this has now become standard of care in our unit for people who choose to do it. Not everybody does. Just one quick summary slide. Hospital administration, the time burden for patients was between about 4 to 12 hours per cycle, which equated to 1 to 3 hours per injection. The vast majority of that, I will be quite honest, is usually sat in our waiting room trying to get into the clinic. But for the self-administration model, that equated to about 2 to 4 hours of people's time per cycle, which equated to around 30 minutes per injection, and that would be at home. This is one of my lovely patients in his just came in from the garden, washed his hands, did his bortezomib and went back out again, got on with it. So I've only got 4 slides left. What am I going for time? I can't see my time. I'm probably over, hopefully not too much. 4 slides left. 2 on, I just wanted to give some information around the sort of relapse refractory treatment in myeloma and resource restricted regions. So the lovely Professor Han Quach, a fabulous myeloma specialist from St Vincent's Hospital in Melbourne, spoke in the treatment outside immunotherapy session and talked about some of the common challenges that we have in resource restricted areas. And despite Australia having a universal health system, we do still have significant limitations with access to novel therapies, as you are all aware. So, and some of the common challenges, and this is not just Australia-centric, this is sort of resource-restricted areas, is this restricted treatment options. So there may be prohibitive cost or limited access to therapies, delayed presentation with wide variety of access to healthcare, rural, regional, remote areas, private government funded, self-funding, and presentation of people with advanced disease, which we still find is a problem occasionally. Limited hospital resources with staffing in, centres, in ED, with access to radiology, access to multidisciplinary support, and limited access to response assessment after treatment, such as PET and MRD, which I'll just speak a little bit more in the next slides. And also limited access to risk stratification tools, such as FISH. You've heard about some of these things today, microarray, molecular studies. So there's a limitation on our access to them. And HANG is really good because she brings it back to when you've got restrictions on resources, just get an accurate diagnosis, the best, most accurate diagnosis you can probably get, and give the best available therapy. Sorry, I speak with my hands, they're all over the shop. And just one more slide from HANG, and this was, her approach to choosing the next choice of regimen for patients who are have relapsed myeloma. So she looks at efficacy, so always using the most effective regimen as early as possible. The greatest impact is when the disease is less resistant. And we know that everybody does not make it through every single line of therapy. So, you know, it's always using absolutely what you've got best available next. And the treatment benefits, the deliverability, treatment benefits must depend on effective delivery. So that's, yes, the delivery method and your ability to access treatment, or if you're not local, but also we have to think about that cumulative toxicity, neuropathy, and knocked about bone marrow and cytopenias. So it's important to really think about the whole toxicity that comes with that regime. And maximizing remaining options. So treatment should always prioritize efficacy. Don't save something for later on down the track. If it's the best drug available, you use it now. And she just spoke a little bit about the IRIL study. So that's a very good example of this response adapted approach. So without trying to give everybody every single drug you have available, not everybody needs every single drug you have available. So that is built on a backbone of therapy, and people who failed to respond appropriately then had the addition of isatuximab. Everybody didn't get it up front, everybody didn't need it up front, but the people who responded poorly did. Have I got time for two more, Natalie? I've got time for two more slides. I can see you, so you'll have to answer me. Oh, thank you. Sorry, I get verbal diarrhea. Last two slides anyway. So Dr. Nader Corday is the clinical director at the Memorial Sloan Kettering Institute in New York, and she's fabulous. And she was presenting in the oral abstract session, looking at maintenance therapy cessation for people with myeloma. So as we know, lenalidomide is a cornerstone of myeloma, and it has its issues. You know, I don't, you know, detract from the fact that it has significant side effect profile for some patients, not all. Fatigue can be a problem, diarrhea, you know, knocked about blood counts, financial issues with toing and froing. But it has been an absolute game changer for prolonging remissions and extending overall survival when we think to those pre-maintenance days. There's been a long-term analysis of the UK Myeloma 11 trial, which suggested that continuing maintenance in patients with sustained minimal residual disease. So minimal residual disease means this just a very, very low level of myeloma. So we know the bone marrow is clear. There might not be any evidence of disease, but when you look at an MRD, that's that molecular level, you might still see plasma cells there or myeloma cells there. So somebody who is MRD negative means that you can't see them there either. So the UK Myeloma 11 trial said that, so for these people who haven't got evidence of disease, when we do that deep MRD testing for 3 years on lenalidomide, do they really need to have lenalidomide carried going forward? And a valid question, one that we would all like answered as soon as possible, please, because people don't want to be sat on treatment that does have side effects, obviously. So she presented her updated results from a prospective phase 2 study of len discontinuation. So they had 50 people in their study, and they started to discontinue patients after they'd had 3 years of sustained MRD negative remission. So no evidence of disease at that molecular level for 3 years. And the results were that, yes, this is very feasible after 3 years of MRD negativity to cease lenalidomide. So for MRD negative patients, at 1 year of stopping therapy, 85% were still in MRD negative remission. And for patients who were tested at 2 years, still 78% of patients were MRD negative remission still. So there was lots more analysis, but I think they were just the key points I wanted to say. So it is highly feasible that people do not need to be on lenalidomide forever. We've got people who have been on it for 10 years plus, and they're absolutely wedded to it because they're worried about stopping. What we don't really know is who's it okay to stop on and who's it not. And that's the questions that need answering. And hopefully the SWOG 1803 study will help us along there. So that's a randomized study which will randomize patients to continuing maintenance therapy or discontinuing after 2 years of being MRD negative. And we are all eagerly awaiting what the outcome of this trial will be, because it will hopefully mean we can de-escalate therapy for many patients. And I just wanted, the last thing on there is just to say we don't have easy access to MRD yet in Australia. Hopefully that's something coming. And on that note, I'd just like to say Mutu Obrigado. And this is Tracy King and I, Dr. Tracy King and I, at the ball in Rio in our outfits, following a couple of cocktails. Thank you, Laura.
Thank you so much, Jackie. You really did an amazing job of articulating what I know would have been some very complex work into very easy to understand information for our community. So thank you again. I do have a couple of questions if you have 5 minutes up your sleeve. Yeah. Firstly, a question that we often get raised on the telephone support line and is a hot topic in our support groups, steroids, which, I know you know a lot about and touched on quite a bit there. Acknowledging that each person is unique, and this is very much not tailored advice, in someone who's on steroids as part of their treatment regime, how would you kind of get a bit of an idea as to when and how might be a good opportunity to reduce the dose of the steroids?
Look, and I think you hit the nail on the head it's about the individual in front of you, isn't it? I think we always feel more comfortable de-escalating steroid dose when somebody has a response for a start. So we try and keep, always keep full dose steroid through first cycle, unless if somebody has, occasionally we'll get those really severe reactions like psychoses or a problem like that. But really, I think we like to get people to the stage of response. So that doesn't have to be absolutely a complete response, but we know that over time, the steroid is less important. It's more important at the beginning when we're trying to get disease control. And we'll often keep steroids going a bit longer if somebody's got a lot of pain, for example, because they're very good anti-inflammatory. So there are pluses and minuses with steroid. I always think it depends a little bit more on the severity of the symptoms. So I would encourage patients to record the symptoms that they think are related to steroids and make sure they're having conversations with the treating people in the day units when you go and when you see your doctor. Because I think often steroids will get left on at reasonably high doses unless someone's articulating the real issues with them. And as clinicians, we're often quite good at, oh yes, I'll keep you awake at night. But if that's not just the one night a week and it's impacting your sleep the whole week, which we see all the time, then that's a different kettle of fish, and we have to address steroid reduction a little bit earlier. You may be pushed to stay on a higher dose of steroid if we don't have good disease control and there's just concerns about de-escalating too early.
Yeah, I definitely see that a lot in my practice and hear that a lot as well. Often people might feel like, oh, I don't want to bother my clinician with my sleep, that's not important, or I'm scared to reduce the dose of any of my treatments in case that impacts the outcome that the treatment has. So yeah, keeping that communication open between you and, whether that be the nurse or the doctor or whoever it is that safe person, so important. Now I have a question from our community on the little chat box here. Russell asks, how can the body's natural cortisone production be boosted without steroid supplements?
With great difficulty.
Yes.
I think, as you know, with steroids, the way we use steroids, we try and preserve your normal cortisol reaction. I think, if there's not really an easy answer to that one, there's not a boost that you can have. I think we have to be very careful with people who've been on long-term steroids. We tend to stop and start without any thought, really, because we pulse rather than have slow and long. But we do have to think continuously. And I think how we manage cortisol levels is by not testing them. To be, in all honesty, I'll be quite honest. I think unless somebody has had a real problem where we've had to refer patients to an endocrinologist for support. We try and just modify the dosing of the dexamethasone or the prednisone to reduce the risk of having a rebound cortisol problem. I don't really think there is any ways of boosting your own cortisol, honestly.
Mm-hmm. And I guess just one further question. If you were considering, all of this really hit home to you as a patient listening today and you wanted to go and talk to your hematologist about this, any tips or tools as to kind of how, without the, obviously not everybody has a tool to use, but any tips on kind of how to articulate this in a really easy way to your hematologist?
Yeah, look, I think just keeping, if you don't have a tool to you, I mean, actually there are some around now that are becoming in standard practice, but keeping some notes. And I think, I do consults all day, every day, mainly myeloma, and you're time poor, you're time limited. So I think if you've actually had a little think about it and trying to, like I said, what the thing I liked about that steroid symptom questionnaire was that it pushed people into saying, okay, so what's the most bothersome? So it's going into consults with you. I think you always need, obviously, I've got to have these scripts and I've got to have this pathology request, da, 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 da. These are my top 3 things I need to talk about today. And you articulate that the minute you go through the door. Right, really looking forward to seeing you. Eager to hear my results. I need to just cover these things to do with you before I leave. I think everybody just needs direction.
Yeah, there's something so powerful about a little list to address during the session.
I say to patients, go and get your list out, go on, get it out. And I'll say, I might not be able to, there's actually 20 things on that. We might not get through them all. Which would you like to discuss first? I think it's just, having that. I do think generally, hematologists are very approachable, and the rapport you have with them, because you have a long life experience with your hematologist, you generally have a pretty good rapport. And with your specialist nurses, they can often help address many of the issues. Or if you don't have specialist nurse in your centre, Myeloma Australia, they've got their helpline nurses there all the time. So really just reaching out to people who can help you either articulate or help you maybe just think about some easy problems to solve.
Wonderful advice. Well, thank you so much again, Jackie.
Well.
We very much appreciate you donating your time to the community, and have loved hearing about your time in Rio.
Lovely. Thanks very much. Thanks, Laura. Have a lovely day, everyone.
You too. Now we're going to have a short break now, so go make a cup of tea, and we will see you in a few minutes where Kat will introduce our next speaker, Natalie. Thank you again.
Okay, welcome back, everyone. I hope everyone had a chance to grab a drink and stretch your legs and are ready for the remaining sessions of our seminar today. I'd like to introduce our next speaker, Natalie Tuckey. Natalie is a qualified psychologist and holds a Master of Psychology from the University of Adelaide. She is actively working towards her endorsement as a health psychologist. Natalie's expertise lies in the intersection of mental health and chronic illness management, with a strong focus on improving the quality of life for individuals living with long-term conditions. She is passionate about the role of mental well-being in enhancing the overall care experience for those facing chronic diseases. Natalie is currently pursuing a PhD with the University of Adelaide and Supportive Oncology Research Group at SAMRI, supervised by Associate Professor Hannah Wardle, Dr. Matthew Iasello, Dr. Melissa Cantley, and Dr. Katina Ali. The research is focused on improving the mental health and well-being during supportive cancer care for people living with myeloma and precursor conditions. And so without further ado, I'd like to introduce Natalie.
Thanks, Kat, for the introduction, and hello, everybody. I'm really excited to be here today to be able to share a little bit more about the research that I've currently been doing, and to provide some more background about what supportive care looks like and how mental health and wellbeing is really important in that space. So from my presentation today, I'm hoping that you'll come away with more understanding about what supportive care looks like, how mental health and wellbeing is really important during this part of living with myeloma. And I've got a short wellbeing activity at the end, how we can use our values to help navigate supportive care. So we'll get right into some definitions about what supportive care looks like. Over the decades, there's been lots of advancements in technology and treatments. But what that means is that people are, living longer, which is great, but they're living longer, I guess, with side effects and symptoms of the disease as well as the treatment. So that's why supportive care is so very important. The definition can vary according to different professionals, but ultimately it's about the prevention, which is really important, and the management of the adverse effects of the cancer itself and the treatment. So supportive care is really about how do we alleviate some of the symptoms and side effects of, the nasty things that you're kind of having to live with the treatment and the cancer itself, and ultimately improving quality of life. And for everybody, that's incredibly different and unique. Depending, on your age, your social environment, the things that you value most, it's really about making sure that you're doing the things that are important to you for as long as possible. And it's looking beyond the active parts of treatment. So, if you're not in an active phase, for example, then that's still really important of your supportive care, so that you're still doing the things that are important to you and you're managing well during that period. It's not just about when you're actually having active treatment. And ultimately, it's about enhancing your mental well-being. So get my slides organized here. So in the context of supportive care, I wanted to introduce it's called the biopsychosocial model, which is developed by George Engel, and it offers a holistic approach to the relationship between physical health, so physical illness, and mental health. It's something that health psychologists really focus on because it really acknowledges the interconnectedness between, physical and mental well-being, as well as the social structures in which we live in. It can be really helpful to recognize that cancer isn't just a physical disease, and it's really influenced by the psychological and social elements. And you can see on the model here that encompasses those three aspects, which all overlap, which means they're all interconnected. So to go through them briefly, the biological component we're probably more familiar with, which is anything to do with physical symptoms like pain and fatigue, things affecting your sleep, the physical changes to your body like muscle loss, some of the nasty side effects like diarrhea and constipation and different types of pain, for example. But the biological components also include how you personally react to those side effects and treatments, that might be relating to your age, your immunity level, for example. So it's all those sort of physical biological components, which is really important. The social factors refer to our social environment. So this includes access to healthcare services. We've already heard a few comments today about living in a regional or remote area, or if you don't live very close to where your treatment team is. So having access to qualified professionals makes up a big part of that social element. And that's not just about your hematologist and myeloma nurse, it's also the allied health supports that you might be interested in or really used for your complementary care, for example. And navigating the healthcare system would fall into that as well. And then the last little circle there is the psychological factors, which really relate to, how we experience the disease itself, the emotional effects. And I'll go into this a little bit more later in the presentation, with the ups and downs, which happen with myeloma. There might be, feeling anxious or fearful, but you also might feel, some optimism and hope as well. And these can all influence, I guess, the biological factors in terms of, how you're experiencing the symptoms themselves and how you relate from a social perspective, which I should mention also includes our social support. So, your myeloma support groups, your family and friends, for example, your community. So all of these factors really affect your experience with living with myeloma. And I've got a quote here from someone I recently interviewed, and I'll be using quotes throughout this presentation to really highlight how individuals are experiencing this. I'll try and read it from here. So there's one thing that worries me a bit, because with men, we tend not to sit down and chat. We've got to be doing something. I'm with a group that did bushwalking 4 or 5 times with a heavy pack. And the last time I did that, I really struggled to get into the camp in the evening and I knew something was very wrong. I've now gone back on long walks and I can walk with him for an hour on Friday mornings. I go bike riding with another friend, and at some stage I'm worried that if I can't do those things, then that friendship group will fall away. I still catch up with half a dozen people for coffee every now and then, but I'm worried it won't be the same as regularly getting together with people for physical activity. I suspect it's coming to an end at some stage. So you can see that quote really demonstrates how, the physical aspects of the disease is potentially going to impact that social element, because it was previously connected through, physical activity, and that worry and that, fear of uncertainty is kind of playing out. And that's just one example, I guess, how it demonstrates the interconnectedness of our physical, our social, and our psychological aspects. So I wanted to then focus on this model. This is a supportive care framework presented by Margaret Finch. So this presents supportive care into a few more categories, which I think is really helpful for people to identify where are the gaps. Where are the gaps in my supportive care? What's missing? What are the things that are important to me that I still need support with or want to maintain that support? And just want to highlight that the circle in the middle is you, and everybody is unique and individual. So whilst I'm providing some generic examples, it's really about what's important to you. You can't compare yourself to others, or have a one-size-fits-all in our supportive care. So I guess like the previous biopsychosocial model includes psychological, physical, and social. So I won't spend much time talking about those now, but I did want to highlight the other areas of supportive care. So I'll start with the practical element. So the practical needs, just trying to work out what's on the slide there. So the practical needs of someone living with cancer might be the logistics of getting to and from their treatment. So, whereabouts is your treatment located? How many times do you need to go in to have your treatment? Do you need to be there for a long period of time? Other practical matters like the financial stress of managing healthcare costs or stopping work, for example. So access and navigating the healthcare system. That's really the practical elements of your supportive care. And I've got a quote here to demonstrate an individual who's, it's 2 individuals that demonstrated this. So most of the practices now here are not bulk billing, they're mixed practices. So unless you've got a healthcare card or something like that, you have to pay every time you're going in, which is more and more money. I was driving 80 kilometers to get to my medicine, to get chemo and everything. So you can see here that the financial and the transport element of living with myeloma is creating a significant burden for both of these individuals. The next area that I wanted to focus on was spirituality. And this refers to an individual's, I guess, search for meaning and purpose and connection to something bigger than themselves, often involving transcendence or a belief in a higher power or, a deeper reality. Many people, facing cancer often are faced with their own mortality, sometimes for the first time, and they're having these sort of thoughts about why is this happening? What does it really mean? So some people, for spirituality is an incredible, important part of their their supportive care. And it doesn't have to be a religion. It could be about just searching for meaning or being part of or focusing on sort of a higher purpose, for example. And some examples, I guess, of spiritual practice might be through meditation, prayer, mindfulness, connecting with nature can still be spirituality, and being, connected with creative activities like the arts and music, for example. And I've got a quote here from an individual. So I'm not sure what I believe in that respect, but I do believe very much that there is somewhere else to go because of the things that have happened with when my mother died. I feel like, well, if I'm going to go, I'm going to mum. So I talk to her every day anyway. And the last element I wanted to mention, actually, no, it's not the last one. Another one I wanted to mention was the informational needs. So we hear time and time again that, there's information overload, like a lot of complex, difficult information to navigate. And for many people, this is a huge part of their supportive care, to feel like they can be part of the conversation or to be, actively involved in their care. They really need to understand and digest some pretty complex information. So navigating, the medical terms and the terminology relevant to you is really, really quite challenging for most people. Information, I guess, supports relate to not only the conversations you're having with your treatment team, your doctors, your hematologists and nurses, for example, but it's about accessing, having brochures given to you, knowing where to go online, going to the Myeloma Australia website, navigating their services, joining an online social group and going to a social forum, or even conversations with family and friends. So information can actually come from multiple places, not just your treating team. But I think we can all acknowledge it's a huge part of the supportive care environment. A couple of quotes here to demonstrate this. I guess you know what services you need. I guess you don't know what services you need. It's a very new environment to have cancer, but then also to understand what your cancer means and then how it affects you individually. I had to learn to figure out what's there because there is so much support when you know where to get it from. And the second quote there is, I'm an expert on me. I would like to be a partner on that journey and be listened to and supported on what I need, but also have the expertise available so that the person who knows the most in the room, being the doctor, can have that conversation, but in a way that I understand. I don't want to be talked to in jargon. I don't want to be talked down to. I'm capable of having that conversation. I just might need to have it 2 or 3 times while I digest it, but I want to be supported through that. I think that's a really lovely quote that demonstrates the, wanting to be really involved, but just needing a bit more time to really understand what is very, very complex information. The last element I do want to refer to, and I have briefly mentioned it previously, is the emotional support that people need. There's huge ups and downs, depending on where you're at with your diagnoses, with your with the physical symptoms, for example, and it's a huge roller coaster, and everybody's going to be experiencing it very differently. You can have some really raw, strong emotions like feeling rage and anger, or grief that goes part of it, or you might be feeling quite optimistic and hopeful, and you will have these waves, and it might all be happening at the same time. But I think this is a really important factor with supportive care, and that it needs to be present in all aspects in your conversations with your doctors, with the people around you. So feeling like you're emotionally supported, I think, is an incredibly important part. And I've got a brief quote to mention this. So I guess you have to constantly think, oh, well, there's something else is going to happen. But the treatments don't just come along with no side effects. You have to take stupid steroids and everything around them, and that's just what makes you feel gross and horrible. And so, yeah, there's the next line of treatment, and how's it going to make you feel? What new lovely side effects are you going to have? It's just not fair. I'm angry. So this is a quote from someone that I recently interviewed, sort of expressing how they're kind of feeling about the potential of relapsing. So as I've kind of mentioned through, supportive care frameworks, and I've kind of presented them quite in a simple way, but honestly, it's not a simple process. It's difficult to navigate, and there'll be lots of ups and downs along the way as you navigate these changes, throughout your progression of the disease. And it will really depend on, how much you want to be involved, how much information you want. So there's going to be lots of unique factors a part of this. But one thing that is important is this concept of self-advocacy, which refers to, your behaviors in getting your needs and your values met during this process. So this might look like in terms of, asking more questions or doing more research or, joining some, myeloma support groups or, watching these webinars, for example, making it clear what your gaps are, what are the things that are not going well for you, or some of the things that are going well as well, just to sort of keep that conversation going. And being more involved in your care can have some positives. You can see an improvement in your well-being or physical factors by being proactive, just noticing a symptom to start with and going, right, I'm going to get on top of this before it becomes too much, and sort of feeling comfortable to do that. And we're having a sense of autonomy about your own experience, for example, because you're the only person who knows what you're feeling and what your needs are. So that's really important. However, when I was, speaking to people about the self-advocacy component, this can be a really, really tricky part for many people. This is not something that comes naturally to most of us, in a new kind of, environment where there's lots of terms that you don't understand. So I've got a couple of themes, I guess, that came out of those conversations about what might get in the way of being able to advocate for yourself. Some of the comments were around, my issue's not that important, sort of minimizing the fact that, oh no, it's fine. I can live with this. It's not a problem. I don't want to upset my doctor, which came out a little bit in terms of, I don't want to challenge my doctor or come across as of not agreeing with their approach, for example, if they didn't see it as being a big issue. I don't want to waste my doctor's time. You can see how busy he is. We all know the time pressures of like needing to get information out. And I think the previous presenter, Jackie, talked a lot about going in with a list, but this sort of came out in the interviews about, I don't want to be seen as, someone who's wasting his time or her time. I don't have enough information yet. Feeling like they can't contribute to the conversation because that information's not there. That's my doctor's job. They know more than me about myeloma. Surely my doctor will mention it when the time is right. I don't want to be seen as needy. So these are the comments that came out for people when we're talking about how do you get your needs met. So I guess in terms of the challenges that people are experiencing is, feeling like they need to understand the diagnosis more before they can speak up about their needs, how they engage with their treatment team, the relationship, the rapport they have with their hematologist, their GP and nurses. Making your needs known, sort of feeling like you can do that, being open for asking for help. This can be a huge barrier for people wanting to sort of say, actually, I'm not coping, I'm not doing really well, which is really hard for many people. And then that confidence to speak up in a really unfamiliar, environment that you may not be used to. So this is where I guess mental health plays a really big role. Navigating all these challenges is absolutely complex and can be really hard for many people. And as I've mentioned, bring up a huge range of emotions. So mental health is defined as a state of well-being that enables people to cope with the stresses of life, realize their abilities, and learn and work well. So it's about feeling resilient, that you feel like you've got the skills and the ability to cope with those challenges that are ahead of you. So mental health awareness and education can help us to cope with those emotional ups and downs. And it's normal to have ups and downs, but sometimes they can become too much, or they start to interfere with our normal day, for example. So I'm just going to go through, I guess, 3 simple factors which we should be thinking about with our mental health to keep us mentally well. And the first one is managing our emotions. Like I said, it's normal to have ups and downs, but if you're noticing that you're spending, there's more time of your day where you're feeling really anxious, or you're feeling like you just don't want to do things anymore, the things that you used to do, that's a warning sign that you're not coping and that things aren't going so well, that you actually need some help. So it's what strategies have you got to be able to manage those huge waves of emotions that come and go. The next one here is social connection. So it's really important for us to feel emotionally supportive, feel like we're part of a community, which I think is really lovely from the support groups that you might be part of, to be able to share those common experiences, maybe vent along the way, but feeling emotionally supported by, your loved ones, your treatment team, and being able to still be part of your community and not just be focusing on all the medical side of things. And then the last one I want to mention is about having meaning and purpose. So if your week is consumed by really heavy medical conversations focusing on the treatment, that can be problematic. You kind of want to have a bit more of a balance about, right, okay, yes, I've got to deal with that, but what are some of the things that are actually really important to me? What are the things that I want to spend my time on during the week without focusing just on my physical health? So reconnecting with the things that are important to us is really important. So mental health and well-being, which is an area that I'm particularly passionate about researching, is growing. There's lots of benefits about focusing on your mental health and well-being during your supportive care. Managing strategies about your anxiety and depression, which might be significant, or it might just be mild. Building resilience and coping skills, and many more. My team and I recently were part of a research group which trialed a mental health and wellbeing program in this space and saw some really lovely benefits. So there's lots more research coming in this space, and we know that mental health and wellbeing is incredibly important as you navigate your supportive cancer care. Oh, I think I've changed slides. Oh, I'll go to this one first. So it brings me to my wellbeing strategy. I don't know how I'm going for time, whether I'm over or not, so I'll try and keep this brief. But ultimately, our draws upon the things that I've mentioned about, balancing out the things that are really important to us. And often people will say, I want to spend time with family and friends or doing the things I love. But sometimes making a bit more pointed about, well, what am I actually going to be doing during that period? What are the things that I actually want to be doing? So connecting with your values allows you to be a bit more authentic in your life. And it's a way of sort of clarifying what are the things that are important to me, and step back and review kind of where you're at. An easy way of thinking about it is how do you want people to describe you? What qualities and strengths do you have that you want to continue to use? And I'm going to use some examples to help navigate supportive care, because that's what this is all about. But you can certainly go back and, rewatch this presentation at a different stage and think, well, what are the things that I actually want to be doing? And it comes from a therapy called acceptance and commitment therapy, where, you clarify your personal values and make a commitment to do things in a more meaningful way throughout your life. So it's about stopping, and on the slide there, I should have clarified, these are examples of different types of values, but it's really about how would you describe yourself. So I think I'll go back one slide there. So what I would get you to do, for example, is to pick one value that's important to you. As I mentioned, how do you want people to describe you? And it can help using categories. So with my family and friends, in my relationships, I want to be someone who's caring or compassionate, or I want to be funny, or whatever it is that, makes you whoever you are. And then you might be something different when you're looking at your health, or when you're looking at personal growth or leisure. Leisure, you might be more curious and, creative, for example. So you kind of want to pick a value that resonates with you. And then we create a goal, I guess, around that. So I'll just jump a couple of slides. So some examples I've got here. So a personal goal would be, I want to treat myself with self-compassion when I feel overwhelmed and spend time in my garden, which is something that makes me feel good. So the value there would be self-compassion. Another example here is, I'm going to use the value of courage to tell my doctor how I feel. Because we know that people don't have as much confidence in this space. So using a value like courage means, right, I'm going to work on this, something that's really important to me. And then the last value I've got there is curiosity. I'm going to use my value of curiosity to ask my treatment team more questions and learn more about my diagnoses. So if there's one thing that you take away from today's presentation, it's that supportive care is tricky and you shouldn't have to do this on your own. Asking for help, having the courage to actually say, you know what, I need some help with this, things aren't going so well, is probably the one thing that I'd like you to take away from today. In the big purple letters at the bottom, the myeloma information line would be a great place to start. If you've got a good relationship with your treatment team, your myeloma nurse, absolutely ask them. There are services across Australia which offer free counselling through the Cancer Council, I believe. If you want to speak to a psychologist, you need a referral from your GP to do so. But you can get support from your peer support groups, family and friends, for example, to say, you know what, I actually don't think this is working so well. I need some more help. So really, really asking for that is really important. And then, so I just want to, I guess, end on this slide. So it's kind of a whirlwind through supportive care and mental health, but I really wanted to sort of focus on those elements today. On the slide, there is a QR code to a survey that I'm launching today, which is about understanding more about the supportive care needs for people with myeloma, but also smoldering myeloma, MGUS, and the family and friends of people that support people with those diagnoses is really, really important for us. So if you get a chance, complete the survey, which would be lovely. And I also want to acknowledge the collaborators who I work with. So Be Well Co, SAMRI, the University of Adelaide, and the Supportive Oncology Research Group as well. So yeah, I don't know how I'm going for time. Hopefully I'm not over too much.
Thanks so much, Natalie, for that was such an informative session. Unfortunately, we've probably got time for one question. There has got one question come through. Our mental health and wellbeing is a really complex space, and knowing where and how to manage the resources and where they are available is just as complex. So you've given our viewers some ideas on where to access resources and how to access them. Just wondering, someone's asked about spirituality and prayer. Where do they fit into the mental health and wellbeing components of healthcare?
Yeah, and I think it's a really good point. And I think in the research that I've done so far is that there does seem to be a gap in encouraging people to connect with their spirituality in some way or form. And I think it's almost like a topic that people don't want to get involved in. It's like, I don't want to think about, the next phase of my experience, or I don't want to think about, the inevitable. So it's almost like steered away from. People don't want to talk about it, I suppose. And I think this is a really personal question for people. It's like, if you are a religious person, or if religion and prayer is important to you, well, then absolutely it should be encouraged, and it can absolutely bring benefits, but even if it's not, like I mentioned, it doesn't have to be about religion. It can be about just, being part of, thinking that we're part of a bigger universe. It's not just about the things that we see in front of us and the things that we can touch with our hands, for example. So I think it's a really important part of people's care, and it's just about navigating and finding the right people to talk to about this. I think it can be, looked away from some places, which is fine. And it's about finding people that you connect with and talk to. And like I said, it doesn't have to be religious. Yeah.
Thank you so much, Natalie. And we really appreciate you sharing your time with us today and with our community. If anybody does have any further questions for Natalie, please make sure you pop them in the chat box, and we will endeavour to get back to you as soon as we can. Okay, I would now like to introduce one of our community members and Myeloma Australia ambassadors, Marlo McCarthy. Marlo is a beautiful mum of 2 young men. She's an interior designer by trade and has a very unique and inspiring story of the lived experience with myeloma. Marlo has been instrumental in raising awareness of myeloma throughout the community and has recently become a member of the inaugural Myeloma Community Advisory Board. It's my absolute pleasure to invite Marlo to share her story with us today. Thanks, Marlo.
Thanks, Kat. Thanks so much for having me, and hello to everybody that's watching this. I'm thrilled to be here and spend a little bit of time telling you a bit about my story. My journey with myeloma began in 2014 when my husband, Tony Walsh, was diagnosed with myeloma. He was 43 years of age, and he'd had a virus that he just couldn't shake, and was subsequently diagnosed from there. At the time, our boys were 6 and 7. Tony went on back then. The treatment options were really a stem cell transplant or a stem cell transplant. So Tony went through the process, had his first stem cell transplant, achieved full remission, and then a month after that, he relapsed. It turns out that Tony had an extremely aggressive form of the disease. And I do really want to emphasize that he was really at the top end of the spectrum in terms of aggressive disease. His story is certainly not what most myeloma patients experience, but he relapsed. It was like a firestorm. Came back, he was incredibly sick, incredibly quickly, and really everything that could have gone wrong for Tony did. He had a go at the first daratumumab trial, and he did have a response to that, but he was unfortunately too sick to meet the criteria of the trial, so he couldn't stay on that. So then the next plan was for him to have another stem cell transplant and then a donor bone marrow transplant. And we were really fortunate that his only brother was a match. So we had all of that set up, but unfortunately Tony wasn't able to achieve remission with that second stem cell transplant, so the bone marrow transplant couldn't go ahead. He actually died 17 months after his diagnosis in November 2015. He was 44 and our boys were just turned 9 and were 7. So that was our introduction to myeloma. Prior to Tony's diagnosis, we'd never heard of it. I often speak to newly diagnosed members of the myeloma community, and I'm just struck by how many people still don't know about myeloma, and that's something that really tugs at my heart, and it is what's motivating me to get involved and be part of advocating for awareness. But subsequently, I then obviously got on with the not little task of being a sole parent and raising 2 beautiful boys on my own, and carving out my career, and wellness and health and fitness was a really big part of that for me. I really wanted the boys to see me very fit and very healthy and very well. I needed to feel that way to do everything that was required of me as a single parent and a working mum. Obviously, the grief in losing Tony was profound, but we are very well supported by a beautiful community, and I have to say that life, has been pretty good to us in the subsequent years. Everybody got around us, and the boys have had, quite an amazing childhood, all things considered. And Tony continues to be a part of our daily life. We continue to talk about him every day, and he continues to parent the boys. But then when I was turning 50 and I was feeling particularly tired and, thought maybe I'd hit, menopause, and maybe I was iron deficient, and took myself off to a GP just across the road from work, and said to her, I need you to do my bloods. Can you just check out what's going on here? So she rang me and said, yep, you're low in iron. But for some reason, just intuitively, I felt like there was more going on. So I went back to her and said, can you just send me my blood work? I just want to have a look through it. And I looked through it and I saw all the same markers of disease that I'd seen with Tony's blood work. And I went back to her and said, I'm in trouble here. I need to see a hematologist. And, I think I've got myeloma. And, her response was to say to me, no, that's an old person's disease. So then I took myself off to another GP who I see sort of for the bigger stuff, and went into her and said, look, I think I need to see a hematologist. And she was like, you're absolutely right, you do. And from there I was diagnosed. So I made the choice to go back to the hematologist that had treated Tony. It was a bit of a double-edged sword. I knew that I was walking right back into the environment that, we'd both experienced quite a lot of trauma in that space, but I also knew that I was going to get exceptional care. The hematologist would have a vested interest in my case, and everybody in that caring group would be determined that it was not going to go the same way as it did for Tony. So And another big thing for me was accessibility to the hospital. So it made a lot of sense to go back to where Tony was treated, which is the Alfred Hospital in Melbourne. So straight away, they started me on the standard line of treatment for a stem cell transplant. I had 2 lines of treatment, and then stopped for a month and had my cells harvested, commenced the 3rd line of treatment, and my disease progressed. So that meant that I have primary refractory myeloma, which is not a good diagnosis. And from there, we knew that we were dealing with something potentially as aggressive as what Tony had experienced. I have to say, right from the outset, I've been blown away by how far treatment has come in the 10 years since Tony started, 9 or 10 years since Tony started treatment to when I started treatment. It's quite extraordinary what progress has been made, and it's incredibly exciting and certainly very reassuring in my case. So I started on an immunotherapy trial of iberdomide, isatuximab, and dexamethasone, and had a really good response to that. That was incredibly exciting. I was able to go from progressing on my 1st line of treatment to being the first person on the trial to achieve complete remission. They then wanted to do a bone marrow aspiration, and their expectation, the medical professional's expectation, was that there would be some residual disease lurking, as that's what myeloma does. But they couldn't find any trace of disease. So that then sort of propelled me to stringent complete remission, which is kind of the utopia of remissions. And again, that was just incredibly exciting considering where we'd come from to get that result. And at that point, I wanted to have a very honest conversation with my hematologist, and I had him alone for a moment, and I just said to him, am I going to be here in 10 years' time? And he said to me, only if we do a bone marrow transplant. And I said, okay, let's go for it. And I need to be very clear that my treatment choices, and they have been my choices, I've been very fortunate to have a say in what goes on, but have been really shaped by my personal experience. And I think seeing Tony miss out on that opportunity for a bone marrow transplant, I just didn't want to miss out on. I wanted to take the opportunities that he missed, and have my boys me do everything I possibly can to stay. So, and it was sort of the time when my disease, it was to come back because as effective as the trial was, it was going to come back at some point, and being of such an aggressive nature, it was going to be a lot harder to treat. So whilst I was in such a great point in remission, I just said, let's go for it, and let's go for it now. So I opted to come off the trial, and then I went to have a stem cell transplant. I had 7 days in hospital, recovered from that, and 8 weeks later, I had a donor bone marrow transplant. My sister was a match, which is pretty amazing. There's 25% chance of a sibling being, a match. And I'm on day 42, 100 days of isolation. It's certainly no small process to go through. I'm sort of at home twiddling my thumbs quite a bit at the moment because there's not a whole lot that I can do. There's a bit of an internal war being waged with the bone marrow, trying to make friends with this foreign body that it's in. And then, of course, externally, I have no immunizations or vaccinations, so I'm at significant risk of infection, when I sort of go out into the big wide world. But I think, so that's sort of the summary. It's quite involved. That's what's gone on for me. But really what I think has been incredibly positive, because there have been so many challenging moments, and I've had to work very hard at separating off, my disease from Tony's disease and his experience, is right from the outset, and we certainly did this with Tony's illness as well, was my focus was to develop relationships with those people caring for me. I'm very fortunate that my hematologist has a nurse practitioner that operates, on his behalf every day, and she's always at the end of the phone. But I would, actively, every time I would go in for treatment, actively really engage with the nursing staff, really try and find out about them and their lives and develop those relationships wherever possible. And it's certainly made my experience of going into the hospital a lot more positive. I had people sort of to catch up with, people that I was interested in, relationships, banter, conversations and company. And I like to think it was also a really nice experience for those nurses as well, and those people that were responsible for my treatment. When I went on to the ward for my stem cell transplant, I was blown away to when, over the course of my stay, 5 different nurses came into my room to care for me. And those same nurses had cared for Tony, who spent probably about 10 months on the ward in his 17 months of fighting the disease. And to reconnect with them was absolutely beautiful. I was well able to remember precise moments of beautiful care that they showed Tony. They remembered him, and they took, exceptional care of me on the ward. So there's been some real nuggets of positivity through this whole experience. But I think, one of the, if I had to sort of highlight anything, I think for me personally on the trial, dexamethasone was incredibly difficult to deal with. And I think there is still, my parents are certainly this way with their healthcare, but there can still be a real mentality around being a good patient. And I really, really struggled with the effects of dexamethasone, which so many myeloma patients do. And I kept quiet about it, and I kept quiet about it, and I kept quiet about it. And eventually I said to my hematologist, I don't think I can keep doing this. And his response was, let's just reduce the dose by half. Like then and there, just in that moment, and I'd been holding on and holding on and holding on and thinking that I shouldn't really complain about this. This is just, what I need to do. And from there, subsequently, I was weaned off dexamethasone. And also certainly being on a trial, I think what we need to remember is that these medical professionals are actively learning from us in real time. These are drug trials. These, are experiments. We need to be a voice. We need to speak up about the impacts of these drugs, of side effects, of how we're feeling, of the issues that we're facing. And because, whilst they have data from studies on things like, progression-free survival and that sort of thing, they actually, what it's like to live with these treatments in real time, that comes from us. So I think it's incredibly important that we, have a voice and feel comfortable in feeding back. And because, it's such a complicated disease to treat. I think the focus can often be so much on what the actual disease is doing. And if we can feed back, we're going to have, better outcomes for all patients and more information to pass on to allied healthcare workers. And I think there's a lot we can, self-advocacy, is just so incredibly important. And something else that I had along the way, which was really positive, was IVIG. That really helped. I know a lot of myeloma patients have that, as an additional treatment to help boost, our immune systems. And I think that was a real turning point for me as well in feeling more well and certainly develop less infections, less viruses, and that was a really, that was an opportunity that I'm incredibly grateful for having. So I think that's sort of, I know we're running a bit over time, so I know I'm speaking quite quickly, but I'm trying to, I think overall what I'd like to say is just what a positive experience my treatment has been and how excited I am, even considering the nature of my personal disease, about what's going on in the space. There's so much work to be done, but actually at the time of my diagnosis, I had 6 fractures from my right shoulder down to my right hip, and my spine is riddled with lesions, but I remain pain-free and very mobile, and you know, looking forward to getting past this 100 days and getting on with a bit more.
So much for sharing such a unique experience, and I think, you know, you really demonstrated the individual care that we need to provide to myeloma patients, particularly when you look at your journey in comparison to your husband's journey as well, and also how crucial clinical trials and research is in developing our treatment and helping people to live better and longer with myeloma as well. So thank you so much for sharing. We have unfortunately run out of time for any questions today, but if anybody has any questions, like we've said previously, please reach out to us via the nurse's email or via the chat box, and we will endeavour to get back to you with answers on any questions. So thank you so much, Milo. That was fabulous.
Thanks, Kat.
Okay, so without further ado, I would just like to introduce our final speaker for today, Dr. Kylie Chimes. Kylie is a specialist clinical and laboratory haematologist at St Vincent's Hospital and Austin Health. She completed her specialist haematology training at St Vincent's Hospital in Melbourne and at the Peter MacCallum Cancer Centre. She is currently undertaking a PhD in supportive care and transfusion medicine, and Kylie's research work is part of the NHMRC Blood Synergy Program at the Transfusion Research Unit of Monash University. Dr. Chai is committed to providing the best possible care for her patients. Thank you, Kylie.
Okay, thanks, Catherine. Can I check? Can you hear me okay?
Yeah.
Okay, wonderful. All right. Okay, let me know, because the audio is sometimes goes a bit crazy on my computer. So, okay, great. And hopefully you can see the slides okay as well. Alrighty. So thanks for inviting me to present today. I'll be sort of focusing a little bit on immunoglobulin replacement in myeloma, because that's the topic of my research. But before that, we were hoping to sort of discuss, you know, how does myeloma affect the immune system? How do we reduce infection risk in myeloma? I'll just advance the slides. You know, what are some of the current approaches to immunoglobulin prophylaxis in clinical practice in Australia? And just having a quick look at, you know, are there any ongoing research or clinical trials looking at IG in this space? So I guess firstly, how does myeloma affect the immune system? We often talk about, you know, increased infection risks for people with myeloma. So you might have heard the term immunocompromised. So people with an weakened immune system, including people with myeloma and other blood cancers, are sometimes described as being immunocompromised or having an increased susceptibility towards developing infection. There are many factors for why this develops. You can see on the screen here on the top right, there are disease-related factors as well as patient-related factors, and there are also treatment-related factors as well. So firstly, in terms of the underlying myeloma disease process, you know, why do people with myeloma develop increased risk of infections? I guess firstly, you know, plasma cells or myeloma cells are a type of white cells and part of the immune system. And we need healthy plasma cells to produce useful antibodies or immunoglobulins, or IG for short. And these antibodies are very important in helping to fight infection. When people have myeloma, they make abnormal plasma cells, and as these plasma cells multiply, they don't make normal, healthy immunoglobulin or antibodies. Instead, they make large amounts of a single type of antibody called a paraprotein, and it doesn't function normally, and therefore there can develop something called acquired hypogammaglobulinemia or reduced antibody levels, and that can lead to an increased risk of developing infection. Apart from that, specific types of treatments can also affect the infection risk. So on the top left, you can see the neutropenia and lymphopenia. So neutropenia refers to a low number of neutrophils or low number of white cells, and specific anti-myeloma treatments can often lower the types of the specific white cells for people who receive these treatments. Therefore, when there is less white cells, there may be less cells targeting a specific type of infections. And people can often develop specific types of infections with specific types of treatments as well. So in the bottom left there, in terms of treatment-related factors, things like, how much steroid you might be on, things like having intensive treatment such as high-dose chemotherapy or autologous stem cell transplant can also affect the infection risk. Nowadays, with newer targeted treatments, including things like CAR T cells or anti-CD38 antibodies or other types of more targeted treatments, I think we're still trying to, our understanding of what types of infections people may experience with these newer types of therapies is still evolving as well. In terms of the myeloma disease process itself, so we can also see that during remission, when people might have myeloma under control, the immune system might recover as well, so that infection risk might improve to a certain degree. So what are some of the common interventions that we commonly see to reduce infections in myeloma? So I guess firstly, having understanding what some of the signs and symptoms are of infections is important, and seeking early medical attention and review. So recognizing, what might consist in early infection, so things like monitoring temperature, recognizing that a high temperature over 38 degrees or having a very low temperature less than 35.5 degrees might be an early sign of infection. Things like knowing what symptoms might herald an infection, things like having a sore throat or a blocked nose, or having a burning sensation when passing urine, things like that, may prompt someone to seek an early medical attention. So if access to a nurse practitioner or a doctor at your center would be important. There are also some behavior modifications that can help reduce infection risk. So this might include things like regular hand hygiene, before food preparation, for example, or after being out in public spaces is important. Oops, sorry, I've got to advance the slide. Here we are. And also things like food preparation is important. Things like keeping raw and cooked meat separately as well is important in this aspect. And also bearing in mind, what are some of the things that we can do to reduce the risk of infection? Things like perhaps avoiding crowded public spaces might be important as well, or perhaps using public transport during sort of less busy periods may also be helpful. There are other specific types of interventions that can reduce infection risk. So apart from hand hygiene and social modification, things like prophylactic vaccinations, use of prophylactic antibiotics or antivirals in specific cases, as well as use of prophylactic immunoglobulin therapy may sometimes be recommended by your health practitioner. In general, vaccinations that are recommended for people with myeloma include inactivated vaccines. So things like influenza vaccine once a year, 5-yearly pneumococcal vaccine, COVID vaccines is generally recommended. Also bearing in mind that there are specific types of vaccines, such as live vaccines, that are contraindicated for people with myeloma as well. So we've done some studies looking at prophylactic immunoglobulin in people with myeloma, so I wanted to focus on that for the next part of the talk. So I guess firstly, what is immunoglobulin treatment? So hopefully you can see that. So our body makes antibodies or immunoglobulin as a critical part of the immune system, but immunoglobulins can also be manufactured commercially and prepared from large pools of donated human plasma. And this usually goes through quite a complex preparation process that includes things like fractionation, so separating out different part of the donated plasma, stabilizing the blood product, as well as inactivating viruses. And this can be used to prepare intravenous IG or IVIG, as well as subcutaneous IG or SCIG. IVIG and SCIT can be used in a wide variety of conditions, but for people with myeloma, it can be given once a month or every week, depending on its preparation, to try and replace the amount of antibodies in people's blood. There are also other types of preparations that can be prepared, such as hyperimmune IG, but this is generally used to treat specific types of infections. So I guess, why is this important? So we know that IG replacement can be used for people in blood cancers, and we have specific access criteria as released by the National Blood Authority in Australia. We know that the use of IG has been rising over the last 10 years, and people with blood cancers account for the largest users of IG in Australia. I think there are still lots of variation in what happens in clinical practice, and we're still trying to understand how best to use this blood product to fight infection. Okay, yep. So in terms of how we use this in clinical practice, there was a recent survey that was conducted as part of the ALOG supportive group. And this survey was done probably about 5 years ago now. This survey was sent around to clinicians, so hematologists within Australia and New Zealand, to try and understand, what are some of the treatments that they commonly use to prevent infections. And you can see there that hopefully you can see on the graph on the right that there was a combination of treatments that people commonly administered to prevent infections. So this might include things like IVIG, SCIC, vaccinations, and prophylactic antibiotics. What they also found is that the approach to replacing IG was quite different amongst clinicians, and this might reflect differences in local policies governing access to IG. So, for example, people or clinicians practicing in specific areas within Australia and New Zealand might be more likely to give IG versus people in other areas may be more likely to recommend prophylactic antibiotics, for example. Interestingly, also, there's also been a public summary document released in 2019. So following that survey released by the MSEC committee, whereby they identified that, yes, we do commonly use IG and other treatments to reduce infections for people with myeloma, but there were a lot of uncertainty about how best to use this product. So what we know is that there needs to be further research in specific areas, including when to start IG replacement, how long to stay on IG replacement for, as well as the role of IG in other types of blood cancers apart from myeloma, including things like lymphoma. Sorry, I forgot to advance the slide. So this was the MSEC document there that looked at some of the evidence gaps. Okay, so what I wanted to look at next was really, I guess, just trying to understand what the best evidence for IG is in the current climate. So what we did was a number of years ago, we performed a systematic review at Monash University in conjunction with the ALOG group. Essentially what we did in this review was we looked at, what are some of the best evidence for use of IG, and what are some of the best evidence for use of things like prophylactic antibiotics and vaccinations for people with blood cancers, including myeloma, including chronic lymphocytic leukemia and lymphomas. And we tried to see, does this treatment, we included evidence from randomized controlled trials, which is the gold standard for clinical trials. And we look to see, does using IG, antibodies and vaccinations, does it reduce infections for people on clinical trials? And is there any effect on safety? All right, so when we looked at the evidence looking at IG, antibiotics and vaccinations, we found 21 trials that had looked at these interventions. And interestingly, for evidence on IG, most of the trials were actually conducted quite a long time ago. So for the trials looking at IG use, a lot of the trials were done before the year 2000 and looked at quite small numbers of people. So 370 patients were included on these clinical trials. And when we looked at the evidence for antibiotics, there were more patients included. So over 1,000 patients were included on these clinical trials. And looking at vaccinations, you can see that the number of patients included, that is over 3,000 patients, had looked at prophylactic use of vaccinations. So again, I don't want to sort of spend too much time on the specific numbers here, but what they found was that what we found in the studies on immunoglobulin, again, you can see 8 studies, 7 before the year 2000. The studies on IgUs did reduce the risk of clinically documented infection. So we found that there was a good effect on reducing the risk of infections for prophylactic use of Ig. For the trials looking at antibiotic use, again, we didn't find a reduction in the use of clinically documented infections, and this is for people taking daily antibiotics as prophylaxis. And for the trials looking at vaccinations, yes, we found that there was an effect on clinically documented infections there. All right. But when we looked at some of the trials itself, again, a lot of the trials were published over 15 years ago. So before the and also a lot of the trials were really quite small. So essentially, from the systematic review that we did, we agree with the MSEC findings is that we do need more research in this space to try and understand how best to use Ig now that we have more targeted therapies available. Okay. Next, I wanted to have a look at how we use Ig in clinical practice. So not looking at just clinical trials, but looking at patients with myeloma not on trials in Australia and New Zealand, as well as how we use Ig in other countries. So in this study, again, I wouldn't focus too much on the fine details of the study itself, but I just wanted to touch on how clinical practice might differ across regions. So when we looked at patients with myeloma that were enrolled on the myeloma, so the MRDR, the Myeloma-Related Diseases Registry, we had patients in Australia, New Zealand, Singapore, and Korea. And when we looked at the time of diagnosis to when they started Ig, we found that the rates of Ig use in different countries were actually quite different. And secondly, when we look to see, is there anything that might predict for use of Ig amongst people with myeloma in these countries? I'll show you on the next slide, but in that there were specific types of treatments that were more likely to be associated for Ig as well. When we looked at the use of Ig within states in Australia, we also found that Ig was quite different. The likelihood of receiving Ig was quite different depending on which state you were receiving treatment in as well. So essentially, there is a lot of variability in how clinicians are using Ig in this space at the moment. Which brings me to the next slide. So I just wanted to end with, I guess, a few updates on where we're going in the research space. So, I think we at the moment, we do believe that Ig is beneficial to reduce infections, but what we're trying to understand is, which how is it more likely to be of use in specific types of treatments, and how long do we need to continue Ig for? So clinical trials that are currently ongoing include the Rational Platform trial, which is being run out of Monash University. And then this trial is actually recruiting at several sites across Australia, New Zealand, and Canada. So on this trial, we're trying to understand, how long should we continue Ig for once we've started Ig? And is there any specific factors that might predict for a better outcome with Ig for people with myeloma? There are also a number of sub-studies that we're conducting as part of this trial as well. So as part of this trial, we're also conducting a qualitative study whereby we're trying to understand from people receiving Ig, what is Ig doing for you? Are you having what are some of the barriers that you're facing if you are receiving IG, and what are some of the barriers that you might face if you stopped IG, or is there anything that makes it easier or more difficult to receive treatments such as immunoglobulin and or prophylactic antibiotics? So this trial is currently ongoing, but I thought it was important to include just that we know that there's ongoing research in this space for us to try and understand how best to improve the patient experience for supportive care treatments, including IG and prophylactic antibiotics. So just bringing us to the last slide. Essentially, we know that people with myeloma increased risk of developing infections through a number of factors, and that might include things like disease-related factors, as well as treatment-related factors. And there are some common interventions that are used to improve the infection risk, and these include things like immunoglobulin treatment, as well as vaccinations, and maybe use of prophylactic antibiotics in some cases. But we know that ongoing work is required to improve the understanding of how we're currently using IG in this space, and trying to understand what the patient experience is, trying to improve make it easy for people to receive ongoing treatments such as this, because we know that there's a lot of time required in hospital to receive immunoglobulin treatments and other supportive care. But that's the last slide. So I think we are on time, hopefully, at 2:30. Thank you.
It's such an informative session, and we can see that the use of immunoglobulins is so wide and varied, and obviously will likely, with the research you've shown, change in the future as well. And I know one of your research pieces was about how long someone should continue immunoglobulin therapy for. Is there any current recommendations around that, or is that obviously why you're looking into that with your research?
Yeah, so I think there is this criteria for access to IG that's published by the National Blood Authority, but I think we don't really understand from clinical trial work as to how long someone should stay on IG treatment for. And of course, there's a lot of factors that play into that. How stable is the myeloma? What treatments are they receiving at the moment? I think a lot of the time the decision is made to clinicians to decide on a case-by-case basis, and I didn't really show that in the slides, but that's what we picked up from that clinician survey that was done by the ALG a number of years back, and is that how clinicians took patients off was quite variable. Some people might choose to take them off if they've been infection-free for a period of time, or some clinicians might choose to take people off IG if their disease was stable, or some people might choose to take people off if, for example, the IgM or IgA levels had risen to a specific level. And some people might choose to take people off IG at, in specific times of the year. So say, when it's not winter, for example. So there was a lot of variation in how this is done in the hospital setting. And that's sort of part of the reason why we are hoping to sort of collect more sort of evidence-based information to help guide clinicians as well as patients who may be receiving these treatments for a long period of time.
Hopefully
be safely.
Yeah, it is such a precious resource. So hopefully we have some better guidelines for use in the future. Just one last question as well, in relation to subcutaneous IG versus intravenous IG. Is there any differences in effectiveness in the use of these?
Yeah, I don't think we know. The studies that have looked at both intravenous and subcutaneous have been focused on, patient experiences, and there hasn't been any sort of head-to-head data in the myeloma space as far as I'm aware. I think the thought is that with the subcutaneous replacement, perhaps the levels are more steady because of the weekly injections versus the monthly infusions, but I don't think we can extrapolate from current trials as to which is more superior at the moment.
Okay. Well, thank you so much, Kylie. We really appreciate your time today. And that actually concludes our seminar program for today. So we do hope you found the sessions informative. And those of you who are still watching, you're welcome to send any questions through to our nurses email at nurses@myeloma.org.au, or you can book a follow-up phone call on our telephone support line. We really would love to hear your feedback. So if you do have a minute to complete our feedback survey, just click on the link below the video on the webpage. Before we officially sign off, I'd just like to give a huge thanks to all of our presenters today for their insights and knowledge, and to the organising team at Myeloma Australia for pulling these seminars together in such an amazing program. Thank you also to all of our sponsors. And last but certainly not least, a massive thank you to Liam, Darren, and the whole team at CVP for their skillful production of today's event and the whole Myeloma Month seminar series. We hope you have a wonderful day from wherever you are watching, and thank you for joining us.